Okay, I'm going to start by agreeing with the previous coordinators, saying that it's been really a pleasure to work in this cluster. I think that we all have been an added value for each other. And who knows, maybe we can find the way of continuing in touch and continue working together. So I'm going to present the key outcomes of Plasticheal. In 20 minutes, it's going to be just a selection of a few things. This is more or less the overall view of the project, the same than the others. The idea or the objective of Plasticheal was to obtain new relevant data in support of risk assessment, risk evaluation. This means data in the two main components of the risk assessment. In one hand, this is the exposure, and in the other one, the effects. And also, we have made effort in developing in silico tools to be able to connect all these things together. So if I have to present our achievements at a glance, we have generated in this time 58 scientific articles, 46 deliverables, 6 book chapters, and 3 policy briefs. So there's a lot of information, a lot of data on many different types of plastics, many different models, many different endpoints. So the real challenge here is how to organize all this information in a way that's comprehensive and can really help assessing the risks. So for that, we have developed a risk assessment framework that we call Plastic Risk Cut. This is an example. You have there an example of the potential application. The idea is that in this framework, we have different categories that you see around the circle, different categories that are inspired by the results or the things that we have obtained in the project, but also from guidance and regulation. And then from the bottom, from the middle to the extremes, you have the weight of evidence from weak to very strong. So this is how more or less very quickly how this framework works. And we have managed so far to apply this framework to polystyrene and to nano PET, both nano. Polystyrene are the beads. PET, it's a real material, real life material that we have obtained from plastic bottles. And this is how it looks. I have put these two separate because the level of evidence that we have, or the level of results, the level of publications on one side and the other is very different. And this weight of evidence is influenced a lot by the number of studies that we have. So these pictures may be changing in the coming months or time because we will be increasing the information that with what we are feeding this framework. But this is so far how it looks. It's possible that I have time to specifically look into each one of the categories and explain what is the studies that are supporting this. I'm going to take just a couple of examples. This is the summary of the information about immunotoxicity or immune effects for nanopolystyrene and real-life nano PET. So we have assessed inflammatory markers on many different types of models, from the lower to the higher complexity. And when you say a plus, it's because we have obtained positive results, and if not, you see a minus. You see that there's in brackets human exposed at work, because we have observed positive for inflammatory markers, but of course we don't know if all these workers are exposed exclusively to one or the other. So this will be positive for all the plastics there. And we have a clear information that the nanopolystyrene or the beads activate the inflammasome, as you see there. So that is for us a higher level of evidence than the rest of the endpoints that we have analyzed. Another endpoint that you have seen in the framework is carcinogenicity. We have assessed long-term effects and carcinogenicity in particular, but only using in vitro models. So we have to be cautious on how we interpret this. The summary that you have there is what we have. We have assessed the literature and produced a review with this, where we have sufficient indirect evidence supporting for potential genotoxicity, carcinogenicity. This means genotoxicity, inflammation, and fibrosis, for instance. And then we have assessed genotoxicity ourselves. We have conducted the experiments with the VAS cell transforming assay. That's an OECD recognized system. And then we have also applied our long-term system with the cells long-term exposed to plastics. The weight of the evidence is from weak to moderate because, of course, I think we have enough evidence to see that we need to continue pushing and try to assess for this marker. But again, this is only in vitro data in a limited number of plastics and systems and results. Before we got there, I show you some results about what is the big picture, and now I'm going to the more particular way. Before we got to this big picture, we had to do a lot on development of technologies like the other projects did. We had to develop new test materials. We didn't want to rely only on polystyrene sheets, so we did a great effort to obtain new test materials, real-life materials from real plastics beyond polystyrene, and very focused on particle size lower than 1 micron. Then we also developed the analytical workflows that were needed for assessing exposure, and we have relied on 2 different tools or techniques. One is FTIR for what we call microplastics, and the other one is confocal Raman microscopy for those that are less than 10 microns, even less than 1 micron, so what we call nanoplastics. And then we developed a lot of different, well, not developed, actually, the right way is say we adapted and explored how to apply these models to plastics. And you have there a plethora of different new approach methodologies that we have adapted and adjusted for the study of plastics, from stem cells to established cell lines, primary cells, different barrier models of different complexity, the long-term exposure system, organoids, the wall ex vivo blood, an alternative animal model, which is an invertebrate Drosophila, and also some in silico modeling. All these new approach methodologies have been complemented with information from mice, from vertebrate, a mammal, and also from the case studies that in our case was focused on at work space from recycling factory and a textile industry. So with all these developments that we made, we were able to assess for different effects in the different models. For the effects, we have focused in validated endpoints like genotoxicity of inflammation, but we have also moved forward and tried to set up for new biomarkers that are based in mechanistic information. And we have also assessed the effect of modulatory factors such as physicochemical characteristics, but also co-exposure with other potential contaminants. We have made a lot of effort in assessing kinetics. Always in all the studies, we have tried to demonstrate that the particles was able to be inside the target organ or the target cell that we were analyzing by means of different methodologies, like the ones that you have there. And in some cases, we've managed to apply dosimetry models. And for the internal and the external exposure, as I said before, we focused on industry workers. So I'm going to give you some examples of the development and application of these models. For instance, for the exposure analysis, we have worked with the analytical workflow that you see on the slides. We have basically collected the sample, did a cleanup and enrichment, then we filtered all these materials, and then we assessed the micronanoplastics in the filter by means of the two techniques that distinguish nano and the micro in the other side. So more or less this approach or this workflow is what we use for assessing the exposure in foods, the exposure in the air of the factories, and also in the biological samples of the workers. And this is some examples of the results. This is the micro, concentrated in the microplastics. You see the numbers there in the number of particles, the average size of the particles that we found. And surprisingly, this was among the highest concentration that was reported in indoor environments using similar sampling and analytical methods. The main plastic type that was found in the textile industry was PET, And the main plastic type that was found in the recycling industry was polystyrene. If we move from the air of these factories, which are these results, oh, well, this is a nice video where you can see how the partners managed to reconstruct a 3D image of the particles that were found in the air of these particles. It is PET and PES, the main components in the textile and in the recycling. And it's quite clear by these videos that the structure, the aspect ratio, the surface is really very different from the polystyrene or the PET or the beads that we are testing in the lab. So it's something really to consider. If we move from the air to the biological samples of the worker, I'm showing here a couple of examples. The first is an example of one worker with exhaled wraith condensate. This means the air that this worker has in the lungs, has exhaled in the lungs, is still external exposure because the air is still not inside the body. And you see there that the main component, again, as in the air, is PET. But then when we go to the whole blood, now it's internal exposure of one of these workers. In the bottom part of the graph, you see then that there are other polymers that appear like PP, PS, or Teflon. So this is something also to take into account. What is the level of what is translocated from what we breathe to what's in the blood? We will have, of course, more information when we have all the analysis of all the workers together to get to some conclusions. And very interestingly, there's a fraction between 10 and 30% that is really below 1 micron in these samples. Moving to hazard assessment, this is an example, again, of what we have done with advanced gastrointestinal barrier models. We have assessed several different models of this type of barrier. And it is quite clear for us now that the more it resembles this barrier to the physiology of the organ, the better is the assessment of the hazardous effects or the kinetics. And this is an example of a barrier obtained with induced pluripotent stem cells, where they have been able to generate different cell types in the same culture. And as you see in the graphs that you have at the bottom, some particles are causing an effect and some particles are not causing an effect. So again, this is to reflect that sometimes we try to compare micro nanoplastics as a whole, but each one is really very different from each other. Continuing with these barrier models resembling the physiology of the organs, the previous was from the gastrointestinal tract. This is a barrier from the lung. And it's been quite clear also for us that in this case, that the better the system resembles the way the cells are exposed in the lung, this means in the air-liquid interface, they are not in liquid, they are just facing air on the other side, the better are the assessment of the effects and the kinetics that we observe. And here you have some results with PLA. This is a bioplastic that is supposed to replace an alternative to replace fuel-based plastics. And as you can see here, using this system that moreover allow us to assess long-term, like a couple of weeks of exposure, we see structural changes in the mucous secretion, DNA damage, and inflammation. So again, a point to reflect on whether these supposed more safe bioplastics need to be around without us being quite sure that they are really better than the old plastics. And this is to give value also to another point, which is the omics. All the coordinators have started to realize that it's much better to look for biomarkers that rely on mechanisms of action than relying on the old biomarkers that have been tested in many other different projects. And we did that from the very beginning. We have this as all the data sets of omic data that has been generated in Plasticheal. There is a lot of data sets of different plastics in different cell types. And we have developed a way of analyzing this comprehensively so we can test ask, like the machine, what are, for instance, the common pathways that are deregulated for polystyrene or for PET across all different data sets. And then it gives you comprehensive information to detect mechanisms to detect potential new biomarkers. So one thing that we did with this data set is to ask to the system, can we find some biomarkers with potential applicability to human biomonitoring? Can we find other biomarkers, new biomarkers? And for that, we set several criteria. One is if we want to test these markers in the biomonitoring studies, they need to be present in white blood cells. So thanks to the single-cell RNA-seq, where we can discriminate the different blood cell types and the expression changes in each one of these cells, we specifically asked the machine to retrieve the regulated genes in these white blood cells and the other data sets and for all the different plastics. And with that, we obtain a bunch of candidate biomarkers, the regulated genes, and then we tested in a fraction of our biomonitoring study, in this case, the textile industry. It's only 10 controls and 10 exposed individuals where we tested these 13 candidate biomarkers. And what is really nice about this 10 and 10 control and expose is that, as you can see here, there's a difference in the DNA damage. So this is a validated standard biomarkers between controls and expose. Also oxidative DNA damage. This was measured by COMET assay. And also there is a difference between controls and expose for 5 out of the several inflammatory validated markers that we have tested. So these 10 and 10 controls and expose have differences in validated biomarkers. We still don't know what is the exposure of each one of these individuals. And this is something that will come later and really something that I'm eager to understand. But so far, this is the scenario. And then what for the candidate biomarkers was, as you see there, the names are not here, they are coded. The names of the markers, but you see that out of the 13 markers, there are 6 of them that also make a difference between the controls and the exposed individuals. And the reason why they are coded is because this signature of new biomarkers based on omics is one of the key exploitable results of the projects out of the total 12 CARES that we have, and we need to maybe pay attention to protecting the intellectual properties of these markers. I don't want to finish without really highlighting some of the communication and dissemination, because we have done a lot of effort also to try to go beyond the scientific community and reach many other stakeholders. So one thing that we did is that we developed a resource, for an educational resource, and we went to the schools to really test this educational research. So we have worked with 10-year children, try to raise awareness, to educate them on the micro nanoplastic and health connection. We have also made linkages with the more environmental perspective. So we have run expeditions with the Pelagos Institute that works in the Corinthian Gulf, chasing whales. And we have also helped them with that. And we have collected at the same time samples from the Corinthian Gulf to, in the future, maybe be able to explore micro nanoplastics there. And we have also participated in a documentary. We've done a lot of effort also in trying to keep the gender vision, the gender perspective in the whole communication and also scientific strategy. So we have 3 months ago we did an online conference that you see the title there, and now we are here at the CUSP meeting. These are our two Plasticheal opportunities to really discuss with different stakeholders and with the other projects. And our idea is to collect all the input and bring it to the final conference that we have in a couple of weeks. Registrations are open, so I would like to invite you to join our final conference where we will present our final achievements. And I have here summarized. If I have time, I can go through them, but it's just the idea is that now we have a catalog of real-life materials other than beads. We have defined the kinetics of these plastics in vitro and in vivo. We understand more, and there's more to do, but we understand more what's the hazardous potential of these particles. We have made an effort of going deep into the mechanism of action and found new effect biomarkers. We believe we have analytical workflows that work really well for measuring microplastics and nanoplastics in external and internal exposure. And we have generated exposure and effects human data. We have made an effort of modeling those with by means of PBK modeling, and we have developed a wonderful framework where to evaluate all the data that we have generated. And I don't need to go through this. It's just something that we can leave here just to enable the discussion. These are key priorities areas because the idea of this whole conference was to feed a roadmap and feed a policy brief. So this is our personal perspective from the Plasticheal project to contribute to the roadmap. We already did, but this is just our final thoughts on that. Thanks to the consortium, of course, and thanks to all for your attention.